Hello everyone, we are back with another video. Topic of our today's video is Krebs cycle. We have overviewed this topic many times in our previous videos, but today we are going into detailed discussion of Krebs cycle and understand the changes in the structures that will be produced in its pathway. This cycle was named in recognition of the German chemist Hans Krebs, whose research into the cellular utilization of glucose contributed greatly to the modern understanding of this aspect of metabolism. So it is called as Krebs cycle. Krebs cycle is also called as TCA or tricarboxylic acid cycle, because citric acid has 3 carboxyl groups, and other name of this cycle is citric acid cycle because citric acid is first product of this cycle. The most important question you need to know before the discussion of Krebs cycle is, where does this cycle occur? This cycle occurs in mitochondria. Every cell which possesses mitochondria undergoes TCA cycle, like liver, kidney or muscles. Let's have an overview that how Krebs cycle is linked with other pathways, starting from glycolysis. It was a 10-step process that generated pyruvate molecule. Now there was glucose that was converted into glucose 6-phosphate. This glucose 6-phosphate converted itself into fructose 6-phosphate. After that it was converted into fructose 1,6-bisphosphate. Now this element was converted into 2 compounds known as glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Glyceraldehyde 3-phosphate was used further, and it was converted into 1,3-bisphosphoglycerate. This was then converted into 3-phosphoglycerate, then 2-phosphoglycerate, and then it was converted into phosphoenol pyruvate that gave rise to pyruvate molecules. Pyruvate is formed in the cytosol of the cell, then imported into the mitochondria. Here, it is converted to acetyl coenzyme A and imported into the mitochondrial matrix. The mitochondrial matrix is the innermost part of the mitochondria. We will discuss this Krebs cycle later in this video. NADH and FADH2 produced by Krebs cycle. We are not going into details that how many NADH and FADH2 are produced, because we will discuss it in detail while the discussion of Krebs cycle. But these NADH and FADH2 produced by Krebs cycle are utilized in electron transport chain to generate ATP or adenosine triphosphate. It is an organic compound that provides energy to drive many processes in living cells, such as muscle contraction. Location of this respiratory chain is in a mitochondrial membrane. If you want to know more about glycolysis, electron transport chain and integration points of these metabolic pathways, watch our video on glycolysis, electron transport chain, and introduction to metabolic pathways. Now moving into detailed discussion of Krebs cycle with structure involved in its pathway. To have a better understanding, it occurs in mitochondria. So every cell of body that has mitochondria undergo Krebs cycles, such as liver, muscles, and kidney. Acetyl coenzyme A, as you can see, it is 2-carbon structure. It combines with oxaloacetate, which has 4 carbons in its structure. This fusion takes place in the presence of enzyme, called as citrate synthase, and as well as water is involved. 2 carbons plus 4 carbons equal to 6-carbon structure. So 6-carbon structure is generated called as citrate, and coenzyme A with sulfhydryl functional group is removed. It is not reversible step. This citrate converts to isocitrate, a reversible reaction. This is isomerization step. It means the isocitrate produced has same number of carbons as citrate, but the arrangement in its structure is different from citrate. Can you guess the difference in the structure of isocitrate from citrate? Well, location of OH group, and hydrogen is exchanged in isocitrate. Don't forget the enzyme to catalyze this reaction. So enzyme is called as a cometase. Now in next step, there is a production of alpha-ketoglutarate. It is not a reversible reaction. Is there any change in structure? Yes, there is. Isocitrate converts to alpha-ketoglutarate, which is a 5-carbon structure. It means there must be a loss of a carbon. What's this process is called? It's called as decarboxylation, means loss of carbon, N Ad plus converts to NADH. It means there is dehydrogenation. Name of this reaction indicates toward the enzyme that is also called as isocitrate dehydrogenase. But this step is not reversible. Next step is also not reversible. In the presence of alpha-ketoglutarate dehydrogenase, dehydrogenase again indicates that there is dehydrogenation, means conversion of NAD+ into NADH, as in this step. Succinyl coenzyme A is produced. It is 4-carbon structure. It means decarboxylation or loss of carbon dioxide has been occurred. Coenzyme A with sulfhydryl functional group is also added. So succinyl coenzyme A is produced. It is 4-carbon structure. In next step, which is reversible, succinate is produced. This coenzyme A with sulfhydryl functional group has been removed. Now inorganic phosphate combines with GDP to produce GTP, and ADP steals its phosphate and ATP is generated. GTP again converts back to GDP. Succinyl CoA converts to succinate. In the presence of succinyl CoA synthase, succinate is 4-carbon structure. It's a reversible reaction. Both are 4-carbon structure. Water is also required in this reaction. As you can see, hydrogen of water incorporated in coenzyme A with sulfhydryl functional group, and oxygen of water in structure of succinate. Next step is reversible. In this step, fumarate is produced from succinate. It is also 4-carbon structure as succinate. Only hydrogens are removed and double bond will develop. Because FAD converts to FADH2, it means there is reduction. So in fumarate, 2 hydrogens are removed and double bond is developed. As dehydrogenation has occurred in succinate, so enzyme is called as succinate dehydrogenase. Next step is also reversible. In this step, water is added to fumarate. Malate is produced. It is again 4-carbon structure. As fumarate converts to malate, so enzyme is called as fumarase. In last step, which is also reversible, malate converts to oxaloacetate. Dehydrogenation has occurred due to conversion of NAD positive to NADH in presence of enzyme called as malate dehydrogenase. You can remember this cycle by a mnemonic. Angry cow is angry, sing, silly, funny, melody, out. Repeat the mnemonic with me. Angry cow is angry, sing, silly, funny, melody, out. Total 3 NADH are produced, 1, 2, and 3. Whereas 1 FADH2 is produced. Moving towards regulation of Krebs cycle. In the first step, as we have discussed in Krebs cycle, when acetyl coenzyme A combines with oxaloacetate, an enzyme called citrate synthase is used. Its inhibitors are ATP, NADH, citrate, and succinyl coenzyme A. Why ATP is inhibitor? Because when there are sufficient or more ATP, no more ATP is required by the body, so it gives inhibiting signals, similar in case of NADH, succinyl coenzyme A, and citrate. They also give inhibiting signals. But its activator is ADP, because ADP needs to convert to ATP, so it gives signals of activation. When isocitrate is converted to alpha-ketoglutarate, an enzyme called as isocitrate dehydrogenase is involved. Its inhibitor are ATP and NADH. As we have already discussed, ATP is inhibitor because when there are sufficient or more ATP, no more ATP is required by the body. So it gives inhibiting signals, similar in case of NADH, but its activator is ADP because ADP needs to convert to ATP, so it gives signals of activation. When alpha-ketoglutarate is converted to succinyl CoA, an enzyme called as alpha-ketoglutarate dehydrogenase is involved. Its inhibitor are NADH, ATP, GTP, and succinyl coenzyme A. Now let's solve few questions together. After understanding Krebs cycle, each molecule of acetyl CoA entering the citric acid cycle yields how many carbon dioxide? So the answer is 2, carbon dioxides. How many NADH are produced by each molecule of acetyl coenzyme A entering the citric acid cycle? Can you guess? Let me answer you. 3 NAD plus produce 3 NADH, and number of ATP produced by it are 9. How many FADH2 are produced by each molecule of acetyl coenzyme A entering the citric acid cycle? So 1 FAD produce 1 FADH2, and number of ATP produced by it are 2. Does each molecule of acetyl coenzyme A entering the citric acid cycle produce any GTP? Yes or no? Yes, 1 GDP produce 1 GTP, and number of ATP produced by it is 1. So total how many ATP are produced by citric acid cycle? Let's add them. 9 plus 2 plus 1 equals to 12. So total 12 ATP are produced by citric acid cycle. Have you ever thought why TCA cycle is so important? So, let's discuss the function of TCA cycle. TCA cycle produces NADH and FADH2. Both are hydrogen carriers that lead to formation of ATP, as a result of which oxidative phosphorylation is carried out. Oxidative phosphorylation. What does it mean? Let us break down these words first. Oxidative means produced by oxidation. And what is oxidation? It is either gain of oxygen, loss of electron, or loss of hydrogen. Phosphorylation means addition of phosphoryl group. Now where is this phosphoryl group added? It will be added to ADP and form ATP. The process of oxidative phosphorylation is basically occurring in the fifth complex of electron transport chain. Another function of TCA cycle is that it provides for synthetic reaction, for example conversion of amino acids to glucose. Gluconeogenesis is the production of new glucose molecule from non-carbohydrate source. And what are these non-carbohydrate sources? They can be lactic acid, lipids, and proteins. If we talk about proteins, muscle protein is degraded to form amino acids, some of which are glucogenic amino acids such as glutamate, glycine, proline, and many other. They participate in gluconeogenesis. Glycolysis, under anaerobic conditions, ultimately generates lactate, which can be converted back to pyruvate by lactate dehydrogenase. Pyruvate in turn is a gluconeogenic substrate. Thirdly, TCA cycle produces GTP, as a result of which substrate-level phosphorylation is carried out. In substrate-level phosphorylation, phosphate group is transferred from substrate to ADP or GDP, as you have seen in Krebs cycle. Main difference in the substrate-level phosphorylation and oxidative phosphorylation is that the phosphate group is directly transferred from the substrate to ADP to produce ATP, whereas in oxidative phosphorylation, the energy required for ATP synthesis comes from the oxidation of NADH and FADH2 in the electron transport chain. If you want to know more about oxidative phosphorylation and conversion of amino acid into glucose, watch our video on aerobic ATP synthesis, oxidative phosphorylation, protein metabolism overview, and gluconeogenesis. That was all about today's video on Krebs cycle. Stay tuned to sqadia.com for more precise and knowledge stuff videos.